So let's talk a little bit more about what is science. Scientific inquiry begins with observing, then asking a question. Questioning what you observe leads to hypotheses, a proposed explanation. I think point A is correct because of point B. I think what I saw is happening because. Those general questions can lead very easily to hypotheses. You probably do this without even realizing you're performing a type of scientific inquiry, starting with observation and asking questions. Now, when you have a hypothesis, you kind of guess, you predict, am I right? And then you want to make an experiment, make a test. And you want that experiment to only be testing one thing. If you test too many things, you don't really know what your data says, and it can mess up your conclusion. But you would reach a conclusion after performing an experiment that tests one thing, and that would go back to your hypothesis. And you would figure out if, well, was I right? Was I wrong? Either way, you have information. You have answered some of your questions. You may be also posing more. And experiments have to incorporate controls. So there's two types of situations that you have to establish. You have to be testing your hypothesis, but only a single factor of that hypothesis, or one variable. And you're going to be testing a variable that is a part of that observed phenomenon you're asking questions about. You want to try to have controls to guard against any of the effects of unnoticed variables that you can't account for. And so you want to make sure that almost everything is the same and no other variables or parts of your experiment are changed except what you're testing. So, for example, in an experimental test of a hypothesis, researchers are often going to only manipulate one component and they'll keep everything else the same. What's manipulated, or what's considered the manipulated variable, is the independent variable. And then the result of that independent variable being manipulated, basically the measure or what happens, that's called the dependent variable. The dependent variable depends on the independent variable. Or I could say the responding variable, which is the dependent variable, depends on the independent variable, which is what's manipulated. Another way to remember this is your independent variable is what you, as the scientist, have decided to test. Everything else should be controlled as much as possible, so you may even need an experimental group and a control group. So experiments are not always possible because if you can't control for all these various unknown variables, then you don't know if you're really just testing one thing. So your data is not going to be very good data in that case. So there are some experiments that have also taken place that are now treated as cautionary tales that morally were either ambiguous or wrong, and they created calls to action within the scientific community for scientists to do better and be better. Some examples are the Stanford prison experiment in the 1970s. That was controversial due to how the subjects were treated. The Milgram experiment, where participants were led to believe they were shocking real people, and even though they weren't really shocking real people, they had that expectation, and that wasn't fixed or ever clarified until after the experimentation. And that resulted in things like PTSD and trauma, both to the participators and those experimenting, and aversion therapy, which has been known to cause physical and psychological harm to patients. So you want to have an experiment that is both ethical and not impractical. You want to design it with only one variable being tested, and then you want a conclusion that states whether or not your hypothesis was correct or not, and then analyzes that data within the conclusion. Another important thing about science is it requires repeatability. It requires communication. It's great if you do an experiment and you get this data that maybe could change lives. But if you can't report it, communicate it to others, and have others be able to replicate your data, it doesn't hold a lot of kind of sustainability, or it doesn't hold a lot of weight within the scientific community. Science is absolutely useless unless communicated. If it's kept to yourself, it does nothing for the scientific community or humanity at large. We also want to make sure that the scientific information we put out is valid, that we can perform the experiment again, we can get the same results, that others can perform the experiment again and get the same results. So that's something to keep in mind when you're reading scientific journals. Scientific journals are a means of scientific literacy, but also communication. It's a paper being put out with results for the first time to other scientists. So it may not yet have been repeated. It may not have been checked. It may not have been perused by other scientists for validity. It is a first look at new information. So keep that in mind when you look up scientific journals. Let's move on and talk just a little bit about some definitions. So what is a scientific theory? Scientific theories have been thoroughly tested. They are generally reliable, they're an explanation, they explain a natural phenomena. They're not necessarily proof of a particular phenomena, but they're the best understanding we have thus far, and they are very much trusted. And you could really say a scientific theory is a natural law. It's a basic principle. It's something that has not yet been disproven by scientific inquiry, and usually through repeated experimentation and constant observation, reproducible experiments, and communication. So they are very strong. That includes cell theory, the idea that cells are the basic unit of life, and all cells come from pre-existing cells, and cells that come together of many different shapes and forms make up multicellular organisms. Also have the theory of gravity. These are trusted explanations of natural phenomena. So cell theory was the example I gave. So all living organisms are composed of cells. This actually overturned a long-held theory called the theory of spontaneous generation. So you can see with the cell theory that science changes. Science updates itself. When new information becomes available, when rudimentary and kind of early microscopes started letting us know more about the microscopic world, then we changed what we thought because we had more data, we had more evidence. So cell theory says that all living organisms are composed of cells. The theory of spontaneous generation said that things just come from non-living things. So maggots come from rotting meat, or sediment at the bottom of a lake produces eels, or mice come from straw. Because these were observations people had had. Even experiments have been done. But we now know a lot of those experiments were flawed because they weren't controlling for all variables, they weren't testing just one variable. And thanks to better microscopes, we now have tons of data that support the cell theory. So you could even say the cell theory is kind of moving away from being a theory and more an actual law. But currently it's still considered a scientific theory. So what we see from science is that it illuminates life. It lets us know not only about the properties of life, but how they apply to different organisms, how we can organize those organisms within phylogenetic trees and with types of systemics called taxonomy. And science is definitely a human endeavor. It starts with observation. It starts with accidents, lucky guesses, asking questions. It starts with competition between scientists, curiosity, and it definitely is something that is best communicated because you don't want to keep these scientific endeavors to yourself. You want to share it with the community. That's the purpose of science, is to better humanity. But speaking of accidents, let's talk about penicillin. Just an interesting example of how sometimes you do science accidentally, or sometimes you stumble upon a monumental discovery accidentally. So in 1928, Dr. Alexander Fleming had been on holiday, and before he went on holiday, he didn't really clean up his lab, and he noticed that in a petri dish that had bacteria, because he had kind of just thrown this petri dish of bacteria into the sink to be handled later, it had grown to mold. It had just been left alone. He wanted to go on vacation. He was ready for his PTO. Well, this petri dish that originally had many colonies of Staphylococcus bacteria now had less colonies. There seemed to be an area around the mold that grew on the petri dish that no longer could support bacterial growth. So that made him ask questions. Why is the bacteria dying? Why can't it grow next to the mold? What's in the mold that's preventing bacterial growth? Turns out it was a chemical called penicillin. And that chemical is actually a self-defense mechanism that mold produced, but now we utilize as an antibiotic, something that kills bacteria. And we found penicillin all because someone didn't clean up their lab and they really wanted to go on vacation. There's a lot more to the story, but it is the basics. And you can look up many different stories of scientific discoveries and see kind of the pitfalls and the difficulties and the competition. One really interesting type of competition that happened when it comes to scientific inquiry is finding out what DNA looked like. Not just what DNA looked like, but was it DNA that was even the genetic material? We didn't know that for a long time, a lot longer than you think. So if you do want to go down a rabbit hole, scientific accidents and inquiries is one that's fun to go down. But that's it for chapter 1. Make sure to review, get all those definitions in, make sure you have a basic understanding and foundation, and get ready to look closer at all of these different topics.